or muscle fiber, you will see some components. And first of all, it is covered by the membrane called the sarcolemma. And the sarcolemma, when the muscle fiber is stimulated with an action potential, it is transmitted along the sarcolemma. But the sarcolemma forms these pouches that then dive down interior of the muscle cell, and those are called T-tubules. So that's what allows the action potential to go down into the muscle cell and bring the impulse deeper into the muscle. There's also what's called the sarcoplasmic reticulum, and the sarcoplasmic reticulum is a storage site for calcium. When the action potential or the stimulus is being transferred down the T-tubules, it causes the sarcoplasmic reticulum to release calcium ions. And calcium ions are needed for a muscle contraction to occur. So it stimulates that release. And you can see also the myofibrils, and on the myofibrils are some blue and red lines that are reflective of the contractile elements or protein. Also the nucleus, you can see, and then a cell, muscle fiber also, it has mitochondria. Mitochondria are the powerhouses. They produce ATP when there's oxygen present. All right, so we said that the contractile unit was called the sarcomere, and the sarcomere goes from Z line to Z line, or that zigzag purple line. And the Z line is what holds what we call the thin filament, the actin filament. And in the center of the sarcomere are the thick filaments, or the myosin, and those are proteins. The myosin is held in place there again in the center of the sarcomere by protein, and that is what's called the M line, if you look down in the bottom right. And what will happen as the muscle contracts is the actin will slide over the myosin and will pull in towards, the Z lines will pull in towards the center of the sarcomere. Now we have different zones, bands that we also look at beyond the Z line and the M line, where in the very center you'll see kind of a stripe, and that is only where you see myosin. There's no overlap of actin and myosin. There's myosin only, and that is called the H zone. And where we see the region where we see the overlap of actin and myosin, those are called the A bands. And then where there is actin only, on either side of the Z line, you'll see actin going out to the left and to the right of the Z line. That band is called the I band. And so these bands and zones will change as the sarcomere contracts. As the actin starts to overlap more with myosin, you'll see the Z line get smaller. And as the muscle fully contracts and all the myosin is overlapping with the actin, you will see the H zone disappear. And so it's these the way the contractile units are arranged and how they change, that's what gives us the striated appearance in the muscle. And you can also see to the left the yellow that lays over the sarcomere. That's the sarcoplasmic reticulum that is responsible for releasing the calcium, which then migrates to these proteins for the muscle contraction to occur. You can also see the ring around the sarcoplasmic reticulum with the tubes. That's the T-tubules. And the T-tubules, if you recall, are what bring the action potential deep into the cell. So here again, you can see the different zones and the lines that give the muscle its striated appearance. The M line, there again holding the myosin in the center of the sarcomere. The Z line holds the actin on either side of the Z line. The H zone is where we have myosin only, whereas the A band is a dark band because there's overlapping actin and myosin. So when we look at what does myosin, why is myosin called thick filament and actin is called thin filament? Myosin is comprised of this bundle of myosin molecules that contain a head, neck, and a tail. So you can see at the resting state it shows the myosin, just this thick bundle of myosin molecules. And it's the heads of the myosin that attach to the actin and pull the actin over the myosin. And below you will see what an actin filament looks like. It's called the thin filament. And each of the actin are globular, so they're round balls, and it's almost like a string of pearls, one ball connected to the next on a string. And actin in the muscle is two strands of pearls, if you will, that are twisted and wrapped around each other. And at rest, the actin filament is covered with another protein called tropomyosin. And you can see it just is a strand that covers the actin. And what the tropomyosin does is it blocks the binding sites. So it lays over those binding sites so that the myosin heads cannot attach to the actin binding sites. And so what initiates the tropomyosin to move and expose those binding sites is when the sarcoplasmic reticulum releases the calcium migrates to the actin, and it binds onto what's called troponin, which are these specific sites on the tropomyosin. And when the calcium binds with a troponin, it shifts the tropomyosin, and so it reveals those binding sites. So now the myosin heads can attach to the binding sites on the actin, forming what is called cross bridges, and pull the actin over the myosin and cause a muscle contraction. So you can see on the left there it shows the thick filaments, the myosin, and then the thin filaments, which are the strands of pearls, the actin, and that's where the cross bridges form and pull and cause more overlap. So here's a closer version of what the actin and the tropomyosin look like. The tropomyosin, it's protein, a strand of protein that lays over the actin binding sites. When calcium is available, it binds with a troponin site, shifts the tropomyosin, and then those binding sites are now available to connect myosin and the actin. And so what fuels this whole contraction is ATP, adenosine triphosphate. And in the resting state, myosin heads are energized. They already have the ATP and the inorganic phosphate has already been split and energy has been liberated, and so the myosin is energized. But the thing preventing the contraction is that the sites on the actin are not exposed. So once the calcium comes into the cell, shifts the tropomyosin, those myosin heads cock back, they'll attach to the binding site, and then there's a power stroke where they pull the actin over the myosin. And now it's at a low energy state, and to detach from the actin, it needs another ATP to come along and bind with the myosin head. Then the myosin head will release from the actin site, and once the energy is liberated, the ATP is broken down into ATP and phosphate, then it will energize the myosin head again to form another contraction. And these myosin heads connecting with the actin are not this very orderly fashion that all the myosin heads move at the same time. They're all moving, connecting, and releasing at different intervals. It almost like if there were a group of people pulling a rope and they were all at different sites at the rope, they wouldn't all be attaching onto the rope with their left hand and pulling, and then the right hand. They'd all be doing it at different intervals. Now, we already talked about the membrane transmits the impulse across the muscle fiber, and then the T-tubules are what allow the impulse to go deep into the muscle, triggering the release of the calcium. So what we have is a motor neuron, and at its most terminal portion, the axon terminal, there's pockets where acetylcholine, which is a neurotransmitter, are stored. And so when the impulse comes down, the action potential comes down, it triggers the release of the acetylcholine, and the action potential is transmitted across the synaptic cleft to the muscle fiber. And where the axon and the muscle fiber are in close proximity is called the motor end plate. And the motor end plate contains these receptor sites to receive the acetylcholine. And so that then allows the impulse to be transmitted there again along the sarcolemma, down the T-tubule, the release of calcium binding with the actin, and then we have our contraction as a result of the myosin heads binding to the actin sites. So it's this continuous attach and release that is a muscle contraction. And in the resting muscle, it's inhibited by the tropomyosin covering the actin attachment sites, binding sites. So just to review, our actin is thin filaments. Their orientation is they're connected to the Z-bands. The myosin are our thick filaments, and that they're the ones that attach to the actin. And when we see muscle contracting, the Z-bands come closer together. And the A-bands stay essentially the same. The I-band where the actin, we see only actin gets smaller, and the H-band where the myosin, we see myosin only, can disappear. So this just is a depiction of a relaxed muscle, and then we see a contracting muscle in the second diagram where we see the myosin attaching to the actin, and you see the H zone in the upper muscle is quite big, but as we start pulling the actin over the myosin, that gets smaller. And then you'll see a fully contracted muscle. It's a much shorter sarcomere. The Z-bands are coming together. There's a lot of overlap. Our H zone has disappeared, and the I-band is also considerably smaller.